Bismillahirrahmanirrahim. Assalamu alaikum. This is Madiha Sagir from Department of Zoology, Bank Road Campus, University of Education, Lahore. So, students, today we are going to talk about the taxonomic keys. In previous lecture, we talked about the taxonomic collections. As we are discussing our main topic, that is the taxonomic procedures. So, in taxonomic procedures, first we talked about the taxonomic collections, but in today's lecture, we're going to talk about the taxonomic keys. So, what are the keys? Keys are basically, if I speak broadly or in general terms, then keys provide a solution to something. Keys unlock our locked problems. So taxonomic keys definitely resolve the taxonomy of the particular specimen. A fifth type of the key is the branching key. This is very simple form of the key, and this key is meant for the non-specialist who can easily read this key, and this key actually is very useful in case of the small groups. This key is not useful in case of the large groups. So we can use this key for the quick identification while we are working in the field. How can we read this key? It's again very easy. Start from here, right? With specimen in hand, you have to first look at the abdomen size. Abdomen shorter than thorax, abdomen longer than thorax. If abdomen shorter than thorax, then you will be given these two choices and ultimately the species. And if abdomen longer than thorax, then again you have the two choices, thorax with spine, without spine. If thorax is with spine, then you would have again two choices, with single spine or with two spines. And if without spine, then your species must have a yellow band on the head. So if head with yellow bands or head without yellow band. If our species does not have spines, then what we do, we will be looking for the yellow band on the head. If a presence shows that it belongs to the species A4, and absence of the yellow band shows that it belongs to the species A6. So this key is very easy to read and understand. Okay, this is another type of the key that is known as the circular key. Again, and this key is very easy to read, so it helps the non-specialist especially, and it is also meant for the very small groups. With larger groups, this key does not work, and it is not easy to use with the larger groups. So to need to start this key, you have to start from the center of the key. In all the keys, I have chosen the same example just to make you understand how to read the key. I have chosen the same statements like of abdomen and the thorax, spines, etc., right? I have not changed the statements, because in this way you can compare the different keys. Abdomen shorter than thorax. If abdomen shorter, then again you have these two choices. If longer, then you have these two choices. Again, if with horns, then you have these two choices, and ultimately you will reach to your species. Without horns, again you have these two choices. In case of wings, you have thorax pitted or smooth. You have these two choices, and species G or species H. Without wings, body black, body yellow. You have to be given with these two choices, and species E and species F. So you can see this key is very easy to read. You just need to have to start from the center, and then you have to move towards your respective choices. Ultimately you will identify your given specimen. Box key. This is again very helpful for the field workers. Here you can see a box. Again, the characteristics or statements have been given in the form of the box, right? Start from the downward, because upward is the final identification is given. Start from here. Abdomen longer than thorax, two choices. Abdomen shorter than the thorax, then you have these choices. Head without horns, head with two horns. And in case of thorax with spine, if abdomen is longer, then again you have these two choices, and ultimately you reach to your species. And from thorax without spines, you have these two choices, head with yellow bands or head without yellow bands. Okay, species A4, species A6. So this is also very easy to read. Okay, another type of the taxonomic key is the computer key. As you know that computer has this technology has taken a very vital role in our lives now, especially after these online teaching sessions. You will very get to know very well about the importance of the technology, importance of the computer. So in case of taxonomy, we also make the keys with the help of different programs. We can construct dichotomous keys as well as multi-access keys as well. Okay, we can construct different types of key with the help of the computer programs, and it would be very easy because we can add the different figures, photographs, diagrams of the specimen of the collection to the computer. So what we do, we just need to run a data already present in the computer and with our new taxa. So it is easier and less time consuming to compare the characters of the unknown taxa with the characters of the known taxa using the computer. Okay, apart from all this, delta description language for taxonomy is also useful system of programs. So what is this? This is a system of programs, different programs that we use in our taxonomy. We for recording taxonomic descriptions for processing by the computer. We record our descriptions about taxonomic descriptions, and that with the help of the computer, delta programs key generates different type of keys, identification keys. So it will be more convenient, it will be less time consuming, and it will be very easy to use such keys that we have constructed with the help of the computer programs, because we can add different pictures as well. In case of if we talk about the molecular biology, it is just to give you an example. So there are many different computer programs that have the data of your proteins, of your DNAs. So whenever we extract the DNA, we just run the program and we compare the newly extracted DNA with already present DNA sequences. In this way, these researches are known as homology researches. So in this way, what we do, we can easily get to know our results. So the computer keys are very helpful in taxonomy as. Okay, now the features of good taxonomic keys. What kind of features should our taxonomic keys have so that it would be more useful for us? First of all, your choices given should be very clear and should not be very firm. Why? Because suppose one choice is body is reddish brown, other is body is light brown. Now, if the body color is something in between reddish brown, light brown, then what we have to do? So, to look at the intermediate between both characters, we need to fit our specimen to some couplet now. So, the choices should be given like this way that we can fit our specimen into that key. Secondly, it should be illustrated as far as possible. A good key will refer to figures for each character. If key is good, then not only statement is given, but a picture is also given with that couplet. Why? Because it will be more easy to see the picture that either our specimen has the same features as visible in the picture. This is another good feature of the taxonomic key. Each couplet must contain reasonable number of alternate characters. It must not be too lengthy or too short. Like if we have talked about that thorax is with spines, so we should have a reasonable number of alternate characters. Either thorax is with one spine, with two spines, with four spines, like this. We have a reasonable number of alternate characters. We should not be focused just only one spine or without spine. Right. This is another feature good taxonomic key should have. Fourth point is that it should contain characters which can be seen clearly without dissecting the specimen. Sometimes the person or taxonomist who has constructed the key, he may construct the key that represents the internal features of the organism. So when we have a specimen and we want to identify that specimen with particular key, if the key is given with the internal structures, then one should have to dissect this specimen, then he would know the ultimately reach the identification of the specimen. So keys should have the statements of the character that would be easily identified without dissecting the specimen. It should not contain couplets which are overlapping. If your couplets is overlapping, then definitely you cannot make a correct identification of the specimen. How? If you talk about the different heights or length of the body, then in if first couplet it is given 10 to 20 millimeter, and in second lead of the same couplet is given 18 to 25 millimeter, then what would happen? You cannot fit. You can fit your specimen in both leads if it belongs to if it is a 19 millimeter, suppose, so you can fit into 10 to 20 millimeter as well, and you can fit into 18 to 25 millimeters. So such kind of keys leads to a poor identification of the specimen. Vague statements such as body small, body large will confuse the user, and the taxonomist should clearly state the exact range of size. Again, exact range of size is very important. If you have just given the small and just given the large, small could be very small, it could be in centimeters, it could be in millimeters, and large could be very large. So you need to have, in case of the lengths, you need to have to be very careful. You need to add the proper measurements, otherwise your key would be not enough to identify the correct specimen. Okay, now merits and demerits of taxonomic keys. Everything has a merit or advantages or disadvantages. So taxonomy keys also have the merits and demerits. If firstly we talk about the merits, then of course with the help of the key that has already been constructed, it is very easy and it is less time consuming to identify your specimen in hand, right? And keys also have the built-in checks. How do they have the built-in checks? Suppose if abdomen is shorter than the thorax, if you have given the two choices further, then that two choices does not meet to your specimen, it means that you have done something wrong and you need to back to the previous couplet and read the statements again. So they have the built-in checks. If it talk about the dichotomous key, then they are very useful in case of the we can easily identify the closely related organism with the help of the dichotomous keys, and it shows the interrelationship between different organisms. And dichotomous key, if we talk about the physical characteristics or phenotype of the organism, then these keys are very helpful, because it gives almost every aspect of the physical characteristic in the key. If we talk about the bracket keys, then they can easily run forward and backward. Divisions are also very visible in the keys. So these are a very great advantages of using the taxonomic keys. But if of the keys, then we cannot identify large number of sample. If we have a large number of sample or large number of specimens, then it would not be very easy to run the key for individual specimen one by one, right? So some samples are identified through the process of elimination. First we have to eliminate that is not being coming in contrast with the given characteristics. So then we have to identify the or sample one by one. It is only useful when organism that is being identified was or is a part of organism that are part of the key. So the organism is usually misidentified if it does not belong to the key. This we have taken the example in the simple dichotomous key, that if the organism is present already identified, or that organism is a part of the key which you are running, then it will be very helpful. It will be easy to identify your given specimen. But if that organism is new and not previously identified, or it is not the part of the key that you are using, then it will be worthless to use that key. Sometimes it makes the complex mechanism of organism identification. So if we have taken one wrong step, then we can lead to the wrong interpretations. This is another demerit of the taxonomic keys. The characteristics which are devised to identify an organism might not be a distinct feature of the species already existing in the dichotomous key. The key may not include all variations in the species. Sometimes the characteristics are given, but sometimes there are many physical variations are present in the specimens. So of the same species. So it will not it will lead to the miss or wrong identification. Variations will lead to the wrong identification while using keys. Key may rely on features not present in that season. Sometimes we have the specimen that belongs to the species that is present in our key. But what happened, because of the seasonal variations, that specimen has a different appearance, and that appearance does not match the appearances characteristics that are given in the key. So it may lead to the wrong identification. Again, we cannot only depend on the physical characteristics to identify a specimen, because many things that are due to the genetic characteristics which are not visible in the keys. So this is one of the major disadvantage of using taxonomic keys. Again, characters can be defined for each taxon as just yes or no. And if we talk about the dichotomous key, we don't have any other option to use. Okay, students, this was all about the taxonomic keys, taxonomic procedures. First, we discussed about in previous lecture taxonomic collections, and in today's lecture, we have talked about the different types of keys, different types of dichotomous keys. Thank you so much, and have a pleasant day today. Allah Hafiz.